Fabulous. Thank you so much, Professor Thomas Liu and everyone for joining us. I'm actually quite excited about this talk because I think it's at a very important point, and I would like to learn it really well from all different angles. So just a brief bio about the speaker. Dr. Thomas Liu is a professor of radiology, psychiatry, and bioengineering at the University of California, San Diego, and the director of the UCSD Center for Functional MRI. He received his PhD in electrical engineering from Stanford University, where he developed signal processing methods for detecting ultra-low frequency magnetic fields along earthquake faults. Wow. He then transitioned to fMRI research as a postdoctoral researcher at UCSD. His research has spanned a broad range of fMRI-related research areas, including experimental design and analysis, innovative signal processing approaches, characterization and modeling of the fMRI signal, and multimodal imaging of brain activity, including fMRI, EEG, and MEG, and development of advanced imaging methods and applications. So I think without further ado, Dr. Thomas Liu, please take it away. Thank you for joining again.
Great, thank you very much, Pradeep, for the invitation and the kind introduction. So today I'd like to talk a little bit about noise in fMRI. When Pradeep gave me this invitation, it was sort of to do a follow-up onto the paper that I published in 2016. So in the presentation, we'll touch upon some of the topics in that paper, but really just using it as a starting point. And so I'll present some background information just to help people be on the same page, but really trying to introduce also some newer results that the field is finding and hopefully stimulate some discussion. Then at the end, Pradeep also asked me to maybe discuss some issues of QC and QA. So I'll give sort of my personal experience with QC and QA. So what we'll do is sort of probably the first 75% of the talk is really going to be focused on this issue of what is signal and what is noise in fMRI. And then, once we're done with that discussion, we can naturally proceed into QA and QC, and whoever wants to stay with that can do that. And we can talk about it as much as we like. So just to remind everyone, it's been more than 20 years since the first fMRI in humans was performed. And this is a figure from Ogawa's paper in 1992. Obviously, there were three main groups that found that. And it wasn't too long before Weisskopf and others in 1993 started pointing out, that there's a lot of signal, the fMRI signal looked like it was noise. And so in this case, it was at the frequency of cardiac pulsation. So there's been a very long history of noise in fMRI. So typically the way we look at fMRI is, we might look at a grayscale plot, where here, this is a paper by Jonathan Power and others, who really has sort of pushed forward this idea of using grayscale plot. So you can really see sort of across space and time, sort of the whole fMRI signal. And what you'll notice is that if we look at things like head motion or something called the global signal or respiratory activity, there's clearly some correlation between these, what we call nuisance terms, things that we believe are noise, and the actual fMRI signal that's measured. Here you see that with the respiratory belt, there's clearly a lot of relationship between the breathing activity and these dark sort of bands of activity in the fMRI signal. So there have been many observations, and people are continuing to make observations of how physiological measures or things like motion seem to find their way into the fMRI signal. And so we want to understand a little bit about that. Typically, the way most analysis programs deal with noise is within the framework of the general linear model, which is pretty much how most fMRI is analyzed. And so, for example, if you have a task-related fMRI signal, a paradigm, here a block design, you would model your paradigm with your typical design matrix, either an FSL or AFNI or whatever software you use. And then you will model all the noise terms as regressors in some structured noise matrix. And then you would typically just fit this general linear model. And so one of the points I want to make is that by writing the model this way, we've sort of assumed a very sort of hard division between what we care about, which is the signal of interest, and the noise. This example, in some cases, this makes a lot of sense. Over in this case, this is a technique done with arterial spin labeling fMRI, which is a way of measuring blood flow. So it has much lower SNR than the typical bold fMRI. And the idea is that here's the signal you're measuring, which looks incredibly noisy, and here is the paradigm you care about, which is this block design here. And so with physiological noise, you can sort of identify regressors, and then you can remove the noise, and you can see sort of a much better correspondence between the cleaned up signal and the paradigm of interest. You can do another method called CompCore, which is more of a data-driven method, which we found does a slightly better job of identifying nuisance terms, and you can really clean up that signal. And so there is, for especially low SNR cases, cleaning up the noise, physiological noise and other noise is really a very important part of task-related fMRI. And it's pretty clear when you've succeeded because you get a much better agreement between your signal and the signal of interest, or the paradigm of interest. Now, it's less clear when you go to resting state fMRI in terms of, no one really knows what resting state fMRI is. And so when you try to clean up the signal, it's really not clear whether you're taking out signal from noise. And so I'm going to spend a lot of the talk talking about that. But before I do that, I just want to point out this sort of interesting result we came across sort of by accident, which is that even from a mathematical point of view, what you can do with resting in terms of nuisance regression is really quite limited in terms of cleaning up the effects of nuisance terms on both static and dynamic functional connectivity measures. So I'm just going to sort of give you the general result. Of the details are obviously in these papers, but the idea is this, that the blue here is showing a dynamic functional connectivity measurement. So this is like a windowed functional connectivity over time. And then here in black, you're seeing the norm, or the length sort of in every window, how big is the white matter signal. And you can sort of see there's a very strong correlation between your dynamic functional connectivity estimates and your white matter regressor. So you think, okay, no problem. I will just regress white matter out of my signal, and you can do this in many different ways. But the surprising thing is, even after you do that, you end up with the red. Either red or green are the sort of the two ways you could do it. You could either regress out block by block, or you could regress out over the whole time series. And you find that even if you do that regression, you're still highly correlated with the white matter sort of energy. And so the question is, what's going on? And it turns out that there's actually a theoretical explanation for this. It turns out that when you do any sort of regression and then you do the correlation, you're sort of operating, you have these very hard bounds where you're basically the difference between the post connectivity, which is after you've taken out the regressor, and the pre is limited by this equation. And then this is what that curve looks like. And so most experiments are operating in this regime where the regression is really limited in what it can do in terms of affecting your functional connectivity measurements across time. You can play the same game across different scans. And so this is an experiment where you have maybe different subjects, and you do the functional connectivity on these different subjects. And in here, the blue is showing the variation in functional connectivity across those subjects. The black here is showing the sort of the power in in this case, the motion, the white matter, and CSF all put together. And you can sort of see there's some correlation between that and the functional connectivity measurements. And then the red is showing what happens after you regress all that stuff out. And once again, you're sort of limited. You're sort of stuck in this regime here, where you just sort of the nuisance regression really just can't budge you out of that. So I think that's something that we tend to have this idea that, okay, as long as we collect all this data and we apply nuisance regression, it's going to fix all our problems. The truth of the matter is, for both task-related and also resting state, nuisance regression oftentimes doesn't really do much. I mean, it sort of fits some of the variance, but really you're still a lot of unexplained variance, which it may or may not be neuronally related.
Can I quickly interrupt?
Yeah, please do.
Yeah, sorry. Can you delve into that equation on the top right here? What's N0 on the N and?
Yeah, so that is this very simple equation took like a year to derive. So the appendix of the NALSI 2019 paper goes through it and motivates it through some geometric arguments. The basic idea is, if you assume the signals of interest are like sort of have some plane like this, right? And n would be, well, n is the size of the nuisance regressor. So that's the norm of the nuisance regressor. So this n bar term, oops, sorry, I missed it. This n bar is sort of just the magnitude of the nuisance regressor in each window. And the n naught is our estimate of how much of that nuisance regressor is orthogonal to the subspace spanned by the two signals of interest. So in terms of, with the time limitations, I unfortunately I didn't put in the slides that go through the geometric argument. We can certainly, maybe if we're going to stay longer, I'm certainly happy to go through that for anyone who wants to know. But it is sort of a somewhat subtle geometric argument. But the nice thing is there's a nice geometric argument to it, and then the math sort of is what we use to create this curve. So yeah, I'm happy to go through it in more detail if people want to stay afterwards. Great,
thank you.
Okay, the main thing I want to talk about this. So this is sort of a figure from the paper, and I want to use it sort of as a framework for talking about and how we view the noise in fMRI. And then the idea is that this is the paper in 2016, and at the end of the talk, I'm going to give you sort of my updated version of what I think this figure should really look like. The idea is that typically, as I've said before, we take these regressors such as head motion or cardiac activity or respiratory activity, and we treat these as regressors. And a point that was made, I think the first notice I could tell about was this paper by Cabell and Hansen in 2011, which is really just cautioning us to realize that the brain is sending out the signals that are causing the head motion that are somewhat affecting cardiac activity and that are also potentially affecting respiratory activity. And so really, when we've made this very stark division between activity of interest and these nuisance regressors, we're sort of making an arbitrary division. And so this picture is trying to remind us that really a lot of what we're considering nuisance terms is due to brain activity. And so we maybe need to be a little cautious in terms of throwing out the baby with the bathwater. So I'm going to go through a little bit of background. So part of this will be somewhat educational, just to get everyone on the same page. So the basic signal model we would have is we would have that the bold signal we measure has both transverse magnetization at echo time of 0, and then that relaxes with the relaxation constant of R2 star, and we measure it at some echo time TE. The math is in the paper, so I don't need to go through that. The bottom line is, if you look at the percent change in bold signal, which is typically what most people are interested in, you find that there's a magnetization term. That's a delta S naught, how much this term has changed. And then you find there's a relaxation term, the very common minus TE times delta R2 star. So this is very well known to people in fMRI literature. So we're going to take a look at a few examples of this. So the magnetization term reflects any variations in this can affect your fMRI signal. And that's reflecting temporal changes in initial transverse magnetization that's created every time you excite the magnetization at the start of each repetition. And so these are some of the sources we'll go through. And there's a relaxation term, which is going to be a little more, delve into more in terms of what are the potential sort of causes of this. And this is any variations typically in delta R2 star that are going to occur. And this is typically what's of interest for most fMRI experiments. We're looking in changes in delta R2 star that are related to changes in neural activity. T is determined by us, so that's not going to change. And this delta n is sort of all the other terms that we really can't explain, including thermal noise. So this relaxation term is going to, its effect comes through changes in this delta R2 star term. And so we'll look at some of the sources of that. And they typically, these are the sources that will come through, are modulated through modulation of things like CBF, which is cerebral blood flow, CBV, which is cerebral blood volume, and CMR2, which is the metabolic rate of oxygen. Okay, so let's just review that a little bit. So if we look at two signals that are required when there's a change in the initial magnetization, so those two signals would look like this. If I take the difference, because it's a difference of two exponentials, it stays exponential, okay? And then when I divide this by the baseline, which in this case I guess would be x, then I'm just going to get this straight line here, okay? So the nice thing is when we look at percent change, magnetization term is independent of echo time. At this point, this figure was from Prantic-Kunduz paper, which nicely sort of goes through this whole echo time dependence. On the other hand, if I have a change in the delta R2 star, then obviously the relaxation rate changes. If I take the change in signal, it's no longer going to be exponential because it's a difference of two exponentials with different time constants. So I get this very classic difference in exponential curve. Now, when I take this and divide by the baseline, I end up with this well-known sort of linear dependence on echo time. And so we can use this to sort of differentiate what is coming through relaxation term and what is coming through magnetization term simply by looking at whether an artifact, what artifact's dependence on echo time is. So a very nice example of that is the multi-echo ICA method introduced by Prantic in 2012 and then later developed in other papers. And so the basic idea is that for these measurements, you acquire data at different echo times, in this case, 15, 39, and 63 milliseconds. And you do some processing, including some PCA and then some ICA. And the ICA is trying to break the data into independent components. And once you have those components, you can ask and say, okay, which of these components has a dependence on echo time? And those would be in the relaxation term. And then you can ask which of the components are fairly independent of magnetization term, and those would be independent of echo time, and those could be the magnetization term. And so here you have a component that its ICA component has a dependence on echo time, and so you would call this part of the relaxation term. And then this one is fairly independent of echo time, and so you would call this a magnetization term. So it's a really very elegant way to split the data. Part of the challenge in using this is there's a lot of stuff that's in the middle, which is sort of part relaxation and part magnetization, and that becomes sort of the devil in the details of this method in terms of what do you do with all that stuff. But it's still a very nice method, and I'll show you some ways that it's some really examples where this approach has been very useful. So this is looking at two subjects. Clearly there's some motion here, a lot more motion here. If we use dVARs, so a measure of motion here, you can sort of see that there's quite a lot of motion. And then in this case, that dVARs is repeated here in black, okay? And the red is the low kappa term, so the magnetization terms. And so basically, whenever you have motion and voxels are moving in and out of where they should be, then that's really primarily, a change in the magnetization, both due to just the motion itself and also spin history effects. And then if you look at a high kappa term here, which is a relaxation term, it's really not affected by this, this motion. And similarly here, you have the dVARs and the high kappa term tracking each other fairly well. And then the high kappa term is fairly independent motion. So this has two things. One, obviously it shows you that you can use these magnetization terms to really track your motion. And the other thing is just that this is also a really good approach for trying to denoise motion, right? Because then you can just get rid of these low kappa terms and you're left with these changes in relaxation terms. Another thing that I noticed Valor is in the audience. So this is a paper that Valor published back in 2015. And so this was around the period where multiband was becoming really popular. And so there was a lot of observations that there were a lot of these interesting artifacts in the data that basically had the multiband striping artifacts. And so, one of the ways of identifying that is with multi-echo ICA. And so these are some examples of how that approach can be used to pick out components that are really modulation of the magnetization term and pull out these multiband artifacts. Another thing, if you look at fMRI data, anyone who's looked at fMRI data, you notice these drifts, and you're like, what are these? I mean, they're like all over the place, and they vary like greatly from voxel to voxel, and there might be patterns, and you can look at these for hours and try to discern patterns, but really, no one really knows what they are still. I mean, people have different hypotheses, and here, even still, I mean, I think there's still some debate about whether these are magnetization or relaxation effects. Paper by Jennifer Evans from Peter Banatini's group showed that evidence that these were probably non-bold terms. So this drift here is basically a non-bold term, and then the after denoising it, keeping the relaxation terms, you really have a nice way of getting rid of these drifts. So I think that this approach seems to be very promising for very low frequency paradigms, which is what sort of they're proposing. So traditionally, if you're trying to do like a pharmacological fMRI experiment where things are changing very slowly over minutes or hours, fMRI is not really good for that because you have this low frequency confound. In those cases, typically people might suggest using something like arterial spin labeling fMRI, where you have this built-in high frequency, high pass filter. But there is some debate because, in this paper by Lirong Yang and others back in 2009, they looked at these low frequency terms and found that they did have an echo time dependence. And so there is a question about whether really there is still some actual information in these low frequency drifts that you would be taking out with this sort of approach. So I think the story's out. This is probably an area that could take more, could receive more attention, and it seems like it's not settled yet. Another form of magnetization term comes from cardiac activity, just sort of the pulsations that go and cause small movements in brain tissue. These are largely localized to areas around the large vessels and ventricles on the edge of the brain. And once again, you can use something like multi-echo ICA. In this case, this is a magnetization term that was picked out. You really see this sagittal sinus depicted very nicely here. And the last sort of magnetization term I'm going to talk about is something that's actually sort of been known for a while, but it's sort of come back into fMRI in a sort of interesting way, which is it's been well known that for quite a long time, well, since 2001, so over 20 years, that when you breathe, you change the size of your thoracic cavity. And this basically, because of the difference between air and tissue, this causes a different perturbation of the magnetic field, which propagates all the way up to your head. And so this can lead to sort of changes in the actual acquisition, especially for echoplanar imaging, where you're going to have these sort of spurious changes of shifts in the phase encode direction. So that's shown here, where you're looking at the center of mass varying as a function of cardiac activity, and you sort of see this very strong peak at the cardiac rate. And on the other hand, if you look in the readout direction, you really don't see that, because the amount of motion that happens in the readout direction for EPI, that any effects are much more diminished there. So this effect has really sort of resurfaced in the last few years when people were looking at these measures of motion called FD. And what they found was that a lot of these FD measures didn't really seem to correspond to motion. And in fact, they were actually this breathing coming into the causing this sort of motion in the sort of spurious motion in the phase encode direction. And so now there's been a proposal that when you're doing FD, you should really filter out frequencies in the respiratory band to get a better measurement of FD. Otherwise, any censoring approaches you take or any sort of post-processing you do is potentially going to be, you'll be processing based on respiration instead of actual motion. Okay, so now we're going to transition a little bit into looking at R2 star and what are the changes. And just to remind you that what we measure with the bold signal is quite far away from the actual neuroactivity. It goes through this interesting, sort of complicated and still studied pathway that neuroactivity causes changes in blood flow and metabolism and CBV. And all of these things can conspire together to cause changes in how much deoxyhemoglobin you have. And then that leads to changes in R2 star. And then finally, you're left with the bold signal here. Okay. So we want to talk a little bit about things that change R2 star. And I'm going to start about sort of talking about things that change R2 star and were thought to sort of be a very direct modulation of R2 star. And we're going to sort of slowly segue our way into sort of seeing how the picture may not be as simple as we had originally thought it would be. All right. So one of the earliest observations of sort of these low frequency artifacts that would modify R2 star came from Richard Wise and others in 2004. And here, this is the gray matter signal. Okay. And this is the white matter signal. And then this is the CO2 signal. And you can sort of see there is, there is some correlation between these signals. And if you look at areas that are correlated, you sort of see these maps that have been repeated in other studies as well. And so the bottom line is that as you're breathing, you're going to have variations in your carbon dioxide level. Okay, and that just happens based on your rate of breathing. And so the slower you breathe can increase your carbon dioxide levels. If you hyperventilate, you can blow off your carbon dioxide. And so all these things change your carbon dioxide levels. If you increase your carbon dioxide, that's a vasodilator. So for example, that will increase your blood flow, and that, all things being equal, will tend to cause your bold signal to go up. Conversely, if you reduce your CO2, you'll reduce your CBF, and that will cause bold signal decreases. So this effect of CO2 on bold signal has been known for quite a long time. And the other thing it does is it actually, CO2 will actually slow down your bold response. It actually even has effect on the temporal dynamics. So most fMRI studies don't measure CO2, although I think an increasing number are, and that's because it requires additional equipment, calibration. It's not always easy to do. But what is relatively easy to do is to put a respiratory belt around your subject. And the manufacturers have made that very easy to do. So the observation from Rasmus Bern and others was that here is the respiration signal, and then this blue is sort of the envelope, which sort of gives you an indication of what the depth of respiration is and how it's varying across time. And here you can see the fMRI signal, and there is, if you look at it, you can sort of say, well, that probably is some correlation between these two signals. And it's made very clear if you have the person sort of have a constant depth of breathing, sort of cue them to breathe, sort of so that their depth of inspiration is fairly constant. And you can sort of see that really gets rid of a lot of these fMRI signal fluctuations. So Rasmus went on to then sort of characterize the responsive breath hold. And so this is what's known as the respiration response signal, and it has this very classic sort of up-down shape. And so this is a cued deep breath, but in terms of what he found was that actually this could be very useful to take your respiration signal, your RV signal, and if you filter that, then that gives us a much better agreement with the actual fMRI signal that was measured. So in fact, and also gets much better sort of agreement with what the CO2 signal looks like. So for example, I think this is a figure from Chang and Glover. So here you show in black, if you take your respiration signal and you filter it, you get this black signal here. And if you measure CO2, you get this red signal here. And so what it says is that you can actually now have a very easily accessible measurement where you can thereby estimate what the CO2 levels are and then use that as a regressor. So now we're going to start looking at, what RV might be and whether it really is a pure confound, and if not, what we should do with it. So it's a really nice paper. I highly recommend reading it by Jonathan Power and others, published in 2020. It's sort of like an encyclopedic treatment of respiratory artifacts in fMRI, and it sort of goes through everything you can know about respiration and all the different variations. But the main thing I want to point out in this slide is the fact that if you look at measurements of whether it's the median respiratory variation or the mean or the standard deviation, you'll see they change with time. And these are over a 15-minute HCP protocol. So the HCP protocol, in some sense, those long scans didn't really make sense because people were falling asleep. But the nice thing is they actually are a great data set for looking at what happens when people get tired. And so in that sense, it's a great data set for looking at these effects. That might have a less effect on smaller, shorter scans, like 5 or 6 minute scans, which seem to be more common now, but are probably still lurking in the background. So this was the observation that Jonathan Power made. And then what he in this paper also, in this slide also from his paper, he also said, well, okay, if you go back and look at the literature and see what brain regions are most correlated with respiration, you sort of see these very artifacts that have a lot of posterior involvement, a sort of midline involvement. And it's somewhat consistent between studies, okay? And then if you look at papers that go through and look at what's changing in terms of arousal, or what are the components that seem to change over the course of an HCP run, like a 15-minute run as people are getting sleepier, then these are the components that are picked out. And the argument is, if you look at the ICA components or the PCA components or the respiration variability components, these are all things that are changing over the course of an fMRI HCP scan. And they seem to involve roughly different sort of the same brain regions. And so one of the questions is, when we're taking out respiration artifacts, are we really taking out something that's more related to the subject's state of arousal? So now I'm going to sort of change gears a little bit and talk about another nuisance regressor that we really haven't talked about, which is the global signal. And this is something that is not really that important for task-related fMRI, or hasn't been traditionally, but it's sort of been a huge thing for resting state fMRI, and still sort of, I mean, when I started looking at this back in 2009, I figured the field would be done with this in a year or two. It's like we're 12 years later and people are still arguing about it. So we need to talk about it a little bit. So the global signal is very simple. If you're not familiar with it, it's simply, this is sort of a cartoon that sort of gets the idea across, which is you have these different images across time. So these are just a single slice from a resting state scan. And you literally just take the average over the whole brain, or in this case, yeah, I mean, I don't know if this is the whole brain or the whole slice, but the brain actually, if you just look at data, you'll actually see these periods where the whole brain just either becomes very positive or very negative. Okay, there are these global effects that you can see that don't require any statistics. You just look at the data, and in some runs, they're incredibly strong, and other runs, they're not. And so one way of quantifying that is you can just take the mean of all the signals in the brain and then plot it as a function of time. And so this would be the global signal plotted here with these bars. So here, when there's a lot of spatial homogeneity, the global signal goes up in a positive fashion. Now everything is blue and it goes down. And then over here, it goes up again. And so the brain is either very positive or very negative, and in between, it's doing something else. Okay, so that's what you get if you just look at data. You can also play some other games, and you can sort of say, well, what if I look at, so here is in blue is the global signal. And then here's just measuring the fMRI signal in the draining veins, like either the great vein of Galen or the sagittal sinus. And it's a very strong correlation between the global signal and those sort of draining vein signals. But you can go even farther. I believe this was, yeah, this was in the fingertip. And so you're sort of seeing in blue the global signal, and in green is actually measuring oxygen at your fingertip. Okay, and so you see that's very highly correlated. So you have this brain signal that you spent $1 million to measure, and then, or to get the equipment to measure it. And then you can take this very low cost pulse oximeter and get a very similar measurement. And this group, from Blaise Frederick's group in Boston, also went on to show you get similar measurements by just measuring the oxygen at the toe. So these very peripheral measurements seem to also be related to what's going on in the brain. Okay. And the same group went on to show, and this is a point I think we're going to come back to, which is there has been some sort of, in fMRI analysis, there has been some sort of attempt to look at spatial temporal patterns and things moving through the brain. But for the most part, we don't. Even our nuisance regression, typically, we don't really introduce any lags or anything into the nuisance regression, simply because it takes up a lot of degrees of freedom very quickly. But I think there is evidence to suggest that the field probably needs to move in that direction a little more. This is an example just showing how if you take the global signal and you correlate it with every signal in the brain and find out the arrival times, it does seem like this global signal propagates throughout the brain in some fashion that is fairly consistent across subjects. And so, for example, using this diagnostic has been useful for even clinical applications, like looking at delays, blood flow delays across the brain by just using this fMRI signal. So why is global signal still an issue? The main reason is because it's super useful to take out the global signal. So if you use the global signal as a regressor, that's called global signal regression. And what it means is you can go, these are sort of functional connectivity maps on 5 different subjects using a seed in the posterior cingulate cortex. And these are the functional connectivity maps you would get with no processing. If you did some like white matter and CSF regression, also some motion regression, you get this. So even using sort of standard methods, you may not be able to clean up the signal. And global signal regression, for being such a simple method, does a remarkably good job of really cleaning up that signal and making it usable. And just as a side note, even using really sophisticated methods such as the ICA fixed method developed by FSL, it cleans up your signal quite a bit, but it does not necessarily take out global signal effects. Yeah. So just the simple effectiveness of this method has really sort of caused it to sort of hang around for so long, even though there's still a lot of debate about whether you should use it. I think that debate was really sort of highlighted by these 2 papers by Fox and others in 2005 and then Murphy in 2009. This is one of these papers which I think really causes sort of a lot of excitement in the field, which is showing that there is sort of the default mode network shown here in red, and then the sort of the attention or sort of the network shown in blue here. And the main story that they got across was that these are sort of these 2 sort of alternating networks where whether you're thinking about sort of interior activity in the default mode network, sort of thinking about yourself, or thinking about sort of the outside world and using this other network here. But the point that Murphy and others made was that you only get this if you do global signal regression. And so the idea was, if you don't do global signal regression, then you don't really get these 2 nice networks. You don't see these anti-correlated networks. And so that was a debate that raged for a few years. So where we came into the picture, one of our sort of observations was that, this is the functional connectivity map before global signal regression, and this is after global signal regression. And sort of a cute finding we found was that if you gave people caffeine, you could sort of largely do what we call natural global signal regression. It isn't quite the same as global signal regression, but it accomplishes many of the same effects as global signal regression, including sort of enhancing this anti-correlation. Okay. And so you can sort of see that, this is the global signal amplitude before and after, and there's a clear decrease in the global signal, but just by giving people caffeine. So, sort of one takeaway could be you should just give all your subjects caffeine, and then you don't have to worry about this. Another sort of interesting finding we found is that, so here is sort of a functional connectivity map before global signal regression. Here is one after global signal regression. And global signal regression is sort of like this, it's straightforward, but you're still calculating a mean signal and then regressing out every voxel and then calculating the correlations. And so even though it's simple, it's easy to get confused. So we are interested in, is there just a simpler way of doing global signal regression? And one thing that you can do is you can sort of just do what's called global signal censoring, where you can just sort of say, well, everywhere the global signal is high, I can just get rid of that data. And I'm just going to keep the points here in red. So these are the points you'd retain after censoring, and you can change the censoring level depending on the global signal amplitude. And then the idea is all the points you have left, you can calculate the correlation of those. And there's no mathematical issues, right? Because you're just saying, well, what does my correlation look like in all the leftover points? And the interesting thing is you basically get maps that look pretty much like what you get with global signal regression. So in a way, a simpler way of looking at global signal regression that's going to get a little bit at sort of, what it is the fact that you can just get similar effects by just censoring and saying, what the global signal is, that there is this very large activity that is oftentimes obscuring what's going on in the background. And if we just sort of only look at the data in between that activity, we can sort of see what's going on in the absence of that large activity. And so this led us down this route where we said, well, we really can't answer these questions with fMRI alone. So we went down this sort of path of getting simultaneous EEG fMRI experiment data, which those of you who have done that know that this is not for the faint of heart. It's a pain in the butt to do, but we had no other choice. We felt we had to do that. Okay. And what that allows you to do is now you can get sort of EEG spectra, and you can look at, for example, alpha activity, what's happening with alpha activity. And alpha activity, especially if you normalize it by activity in the theta and delta bands, is sort of a good measurement that's been used in literature for vigilance. And you can sort of see here, the surprising thing we found is that in certain subjects, when you give them caffeine, the EEG spectra hardly budges at all. And also the functional connectivity maps don't really change at all. In other subjects, they start off in this sort of low arousal state where there's very low alpha. And then when you give them caffeine, the alpha appears and you end up with this sort of very nice functional connectivity map. And so it actually turns out that this is a very strong effect across subjects. If you look at the change in their vigilance as measured with the EEG, you can use that to predict incredibly well how much their global signal is going to change. Okay, so this was sort of one of our first clues as to, okay, what's really going on here is that this global activity that seems to be present everywhere is driven in part by changes in arousal or vigilance. And you can see this across subjects as well. So for example, certain subjects before caffeine just tend to be alert. Other subjects are drowsy, and other subjects are sort of in between. And where they lie on their global signaling amplitude is also correlates with how vigilant they are. And so this is sort of a naturalistic, without just across the population, that variation. And so now I'm going to try to start tying together different things. So we've talked about sort of things in sort of separate, we've talked about like RV, we've talked about motion, we've talked a little bit about global signal, we talked about EEG, fMRI. So this is a study that I think is a very important study done by Yuan and others in 2013, where they basically showed that if you looked at RVT, shown here in black, and you also measure with EEG, fMRI, the global field power, and here in the alpha band, you see a very strong correlation between those two. Okay, so suggesting that breathing and arousal states, in hindsight, it's very obvious, right? I mean, you sort of know subjectively that that's the case, but in terms of how it affects fMRI activity, it was sort of really not known at this point. And similarly, found that this effect is much stronger when the eyes are closed versus when the eyes are open. So the good thing is most resting state fMRI scans are now done with eyes open. So maybe this effect isn't as big as we thought, but if you're doing a long scan, you're generally going to go from eyes open to eyes closed somewhere within your scan, and depending on how tired the subject is. So I think it is something that we need to worry about. I think another thing that is sort of another paper that just came out last year was that transient arousal modulations contribute to resting state functional connectivity measurements, such with head motion parameters. And so what they found was that, a lot of times you look at these head motion parameters, and then there might be a big effect on the fMRI signal, and you think you're regressing it and taking it out. But what they found was that a lot of times it was really sort of, the evidence was suggesting it was really the arousal changes being co-modulating the motion. And so that, when you're trying to censor these areas out, you may be censoring out arousal areas as well, or out moments of arousal changes. And then finally, I'm going to sort of end with some very recent results from the group at WashU and also the group at Penn State that I think suggest that we may want to look at these, that first of all, there's a lot of information in these nuisance terms, and that we may need to start thinking about them a little differently in terms of how we analyze our data and even how we analyze fMRI data in general. This is a really nice study that which I think is in Science Advances, or one of the the science papers, science journals. So what they did here was they literally just took the RV signal and they correlated it with different areas of the brain along sort of the principal gradient of functional cognitivity, going from motor visual to frontal parietal default mode. And so these are shown in these different colors. And what they find is there's a very strong correlation around, with a peak frequency around 0.03 hertz. It's a very low frequency fluctuation, roughly a period of about 40-second period, which also correlates with some previous findings by other groups. The interesting thing, though, is that they found that this is very organized. If you look at the phase between RV and the brain signal, that it's very organized across this principal gradient direction. And so what you can do is you can sort of find that there is sort of this propagation of RV effects across the brain, where if you're looking at the phase shift, it's very well organized across the brain, across this principal gradient. And in fact, if you look at the principal gradient coordinates as defined by Margulis and others, you see a very high similarity between these principal functional cognitivity gradients, which are sort of moving from sensory areas to higher order areas. And that seems to track that fairly well. So it does appear that a lot of this, the argument is a lot of this global activity, it's not just that the global signal is happening in the same phase all over the brain. It's just that this sort of traveling waves of activity moving across the brain that are causing a lot of this globally coordinated activity. And so their claim, which they make in the paper, which is, whether you agree with it or not, I think is a very interesting claim, is that it's really these spatial temporal variations in arousal that are causing these waves of activity to move across the brain. And that they show in their paper that that can sort of lead to a lot of the structure that we see in functional cognitivity networks. And so it may not be that there's the traditional picture is that you have these different networks and they're talking to each other and they're segregated by function. What they're arguing is that it may be that a lot of that data, especially if you don't take out global signal effects, is maybe just due to there's still organization, but the organization must be viewed within the context of these traveling waves moving across different functional areas. So that was a finding that was published this year. And then a very similar finding was published in cerebral cortex by Xiaolu's group at Penn State, where here they used the global signal and some interesting analysis methods to also view how things were propagating across the brain. And they found very similar profiles. So here they found that the way that signals are propagating across the brain followed, and this is delay from minus to plus. And if you look at the delay profile, it also, once again, looks very similar to this principal gradient that's organized from sensory motor up to higher order levels. They also went on to find that there's actually not just one traveling wave. There seems to be evidence for a wave that's going from sensory motor to higher order levels, and then one from higher order levels down into lower order levels. And so there's this sort of two propagating waves almost that are going on. And it's the ratio of those two that perhaps is related to arousal changes. And so here they're showing that ratio, if they're fairly awake, is down at this level. And then once they go to a low arousal state, it increases quite a bit. And they show this in both humans and monkeys as well. And this is electrophysiological data, so it has none of the confounds of bold data. And they've gone on to show, I think, just a recent paper in bioRxiv, they've actually shown this, that they can see similar things in mice as well, that there does seem to be this overall traveling wave activity that we really have largely ignored in fMRI up until now. So I'm going to end with this is the second to last slide. So previously, I gave you the slide in the background, and what I said was I would probably modify that slide now to have really this arousal effect, right? Because arousal may be one of these key things which is, can definitely affect head motion, respiratory rate, cardiac activity. And so the central role of arousal may be something that we need to start thinking about a little more when we're thinking about, what is noise and what is signal in what we're measuring. And I think even further than that, this is a nice review paper by Azzolini and others talking about sort of visceral signals in the body, from your heart and your gut, and how these can modulate brain activity. And likely these are also related to some of these spatial temporal arousal effects and sort of your sense of self and how that's changing over time. And so even, a lot of what we're trying to get rid of may actually be very much related to signals that are sort of that are affecting our cognitive state and sort of our cognitive perception, even perception of the world is changing and possibly related to these changes in arousal state. So I'll end this part of the talk now, and we can sort of open it up to discussion. And then, as I said at the beginning, Pratibh asked me to talk about some QC issues. And so I have a few slides on that, but I'll stop now and see if there's see if people what people think of what has been presented. So thanks for your attention.
Thank you so much, Dr. Liu. So yeah, I think I can say that we're all pretty much aroused at this point. So, and I have a hand up. We have Tor here. Hi, Tor.
Hi, hey,
Tor.
thanks, Tom. Hi, it's great to hear this stuff. It's really interesting, and I learned a ton of stuff. I'm left at this point feeling somewhat confused about what do I do now, right? So you kind of made the argument: do we control for more things, control for global signal, control for time lagged global signals, and so forth? But maybe they're part of the real, what's really driving processing in ways that are important for us, that like your last slide here, which suggests, hey, maybe you don't want to just exclude that and throw it out, so yeah, where do you come down? Like, what do we do? And maybe regression isn't such a great method for getting rid of it anyway.
Right. That's a really great question. I think we'll have to stumble our way towards a solution as the field does, and we'll probably take some many wrong turns before we get to something. I think one thing is that the first step obviously is awareness and saying, does this really matter or not? And so, for example, there was a paper, I forget, maybe about 10 years old, where they were looking at global signal and schizophrenia, right, and saying whether this is a marker of schizophrenia. And the question is, well, is that really true, or is this just sort of a second order effect of differences in arousal between schizophrenic subjects and those who are not? So I do think that the first step is sort of maybe that we should start paying more attention to measures that might, like RV, or one thing that I think is probably useful, but very challenging to acquire, obviously, it would be great to get pupillometry, in a lot of studies, especially resting state. I think, resting state is really where these things matter. And maybe the answer is we should do less resting state. I don't know, because resting state is so unconstrained, right? So I mean, this discussion can go in many directions.
Yeah. Great.
So I think from my point of view, I think it's sort of there's the arousal piece, but I think there's also the way we analyze data, which has been this mass univariate approach. And I think that works pretty well for certain cases, but I do think that there's these huge spatial temporal activity that the brain is sort of obviously doing quite a lot of. And so it may be that there's new approaches that are needed for that. There was an interesting bioarchive paper I was just looking at. I didn't put it in this talk, but looking at like complex BCA, where they're doing Hilbert transforms and trying to break things into sort of different modes, and that seems very promising. And so I do think that how important it is probably varies a lot from subject to subject and scan to scan. And so it may, if you average enough, it probably doesn't matter, right? But if you are If you're looking for inter-subject differences, or if you really ever want to get fMRI to the point where we can do individual diagnosis, then these things probably do matter.
Yeah, thanks. Just a super quick follow-up, I think, which is one of the ideas I keep playing around with in this space, is to separate out these global signals, but analyze those. And you say, okay, here's a distributed signal that's been pulled out of a bunch of local signals. Analyze that and see if it matters in terms of your task variables, et cetera, or clinical phenotypes or whatever, and then analyze the local signals, because clearly the local signals are really contaminated, right, but by these distributed things. And so it's misleading to try to make a local inference based on something that's influenced very broadly.
Yeah, no, I think that's also a good approach, right? I mean, how much can you explain with the global, like a spatially broad effect versus the local effects? So yeah, I think approaches like that, I mean, just seem to make a lot of sense. I think the challenge we face in fMRI is always the fact that, the beauty of fMRI is that there are so many ways of doing things. And then the challenge is that oftentimes we don't know what other people are doing. And so, so yeah, so I'll have to read up on what so is that in a recent paper that you did, or? No,
I just keep, we've done different things in different papers, but I just keep thinking about this, like what's the sort of solution, right? And this is a sort of a, so it's not published yet. It's just a sort of structural model-based solution that you separate these things out and analyze those. But I hope to write a paper on it.
Okay, well, I'd love to hear more about it when you get to that point.
Yeah,
Great. Okay.
Any other questions? Okay, go ahead, Lars.
Yeah, thanks, Dr. Lieber. That was really a great talk, and I learned a lot, especially about the global signal. So what I was wondering is, when you presented this paper, I didn't really understand the math, right? How much global signal regression really helps the function connectivity. But I'm wondering also in the other assessments that you made, one question that came up also in other talks is, what is the right metric that we evaluate our denoising strategies upon? So basically, maybe it's not the denoising that fails the function connectivity, but the function connectivity measure that is used in that is limited if it's not really influenced by changes in our signal processing, right? Then maybe something is wrong with this measure, this dynamic function connectivity in itself, because it should make a difference how you process your signal to the function connectivity, right?
Yeah, I mean, I think it. Well, I think it definitely makes a difference. It's just whether or not it's the magnitude of the difference is what we're looking at. That it's sort of depending on what regime you're in, it can either make a difference or not make a difference. And so our observation, which was sort of surprising to us, was that it didn't really make a difference. So I do think that, and I think when we do like I think where nuisance regression works really well is where there are sort of massive effects, like where there's a lot of head motion and you censor out an area, or where someone's just, so censoring probably is the most important thing because you're just getting rid of bad data, right? But in general, where there's sort of low level of artifacts, like sort of normal variation in breathing and heart rate and all that, then everything is just sort of at very low levels of variance. When you go back and read these papers, the variance levels explained by these regressors is not very much, right? And so I think that's what we're also seeing, is that really these are more the thing that has the most effect is the global signal, and that's simply because it's a data-driven method where it takes out, it captures so many things, but also it has its issues because you don't really know what you're taking out. So I do think anything that's sort of where you're doing an independent measure, those independent measures tend to have a tend to explain very low percentage of the variance, unless there are huge effects, unless you like really make a person breathe a lot, or they're moving their head a lot, then they're absolutely necessary, and that they can make a difference between having a good map and not a good map. Does that explain your question? I'm not sure I got your question.
I think partly. I do think though that bold itself is also a small signal variation, right? So basically, where you're not on the one hand you're competing with a whole noise floor, but you're also competing with the other variabilities. I think that's exactly the point that you made about resting state versus task based, because we're relying on these fluctuations, and then getting rid of everything that is not of interest is actually much more important than in a task space where it's just all noise, basically.
Yeah, and I think the other challenge comes about is if there really is this spatial temporal evolution, then, you know, from the data we have, I mean, how do you actually map that with enough degrees of freedom to say that you're doing the right thing? So I think for large studies where, you know, you can sort of do the processing and then look at the statistics afterwards, that's fine. But for maybe smaller studies, or even at the, if we ever want to get an individual level, then it becomes an issue, because then you are then trying to say, you know, probably on the individual level, we just don't have enough data to fit the correct models, right? I mean, if we're really going to model, you know, this interaction between the brain and the body and have all these other things, then, you know, we probably don't have enough data.
Yeah, thank you.
Thanks.
Hey, Dr. Liu said he has all the time, so please feel free to ask questions. You know, education is actually a big part of this series too, so don't be embarrassed to ask anything. I actually have a question if nobody else has. Basically, you showed some of these causal diagrams about how complicated the whole process is, from the neural activity to the bold signal that we measure, right? I was wondering, like, I think Tor also quickly commented on it, saying whether maybe regression is not the right way to do it. Both of these makes me think that some of the analysis we are doing are actually is not causal and is just mostly correlational. That maybe actually is confounding the results a little bit, you know? So I was wondering, because I think in multiple dimensions, you mentioned that this is not fully understood. And at the time I was thinking, why can't we build a fully, what you call a causal model of this based on, for example, phantoms or more simplified stuff to better understand this whole process? You know, is this causal diagram fully verified and not controversial, I guess?
Well, I would say that the basics of the model are fairly well accepted. The actual details are still under debate in terms of, you know, there is still a very small field of people working on physiological models of fMRI. And those of you who've been in the field for a long time know, for example, that this thing called like the post-stimulus undershoot, you know, was a very long sort of controversy about that. And the original balloon model, which is sort of still the standard model for this, posited that it was due to sort of slow return of cerebral blood volume. But the latest, you know, and that this was from 1998, so, you know, over 20 years ago. And that is still, for example, the model that is used in the, like, say, maybe SPM dynamic causal model framework. But the latest data, even from the inventor of the balloon models, Rick Buxton, sort of now believes that that's not true, that basically the latest evidence is that it's more due to maybe a post-stimulus dip in the cerebral blood flow that's actually causing that. And so even these basic questions are still not known, and these do have an effect on what model you use. I think the idea is that the part of the reason these models haven't advanced too much is really I don't believe, I mean, there is some support for it in the field in terms of there is interest in the field, but frankly, you know, most of the support in the field has gone towards much bigger projects and things where people just want to use the signals and not worry about the models. And so I do think the modeling has lagged behind quite a bit. And but it's not a simple question to answer. I mean, even something as simple as modeling from neural activity, or it's still not simple. It's still very complicated to do, and you know, probably requires more, you know, animal experiments and things like that. And then if you're trying to then combine it with the whole brain-body interaction, then I mean, even that there's still many unknown issues. And so I think we still don't know enough. It's sort of the chicken and egg, right? You need to know enough about the brain to model it, but you're also using the data to figure out what your models are. And so you're sort of bootstrapping your way up.
Right, yeah, Helmut, go ahead.
I have a question, maybe it's a question and a comment, so my question is, you showed, for example, that the regression, doesn't really work. I mean, because we're regressing out something that is probably much larger than what we're interested in the signal. That's probably the reason. And a lot of people have shown actually that logical measures show similar functional connectivities than what we think is like the functional connectivities. And so what my question is and it all boils down to not having a ground truth, right? We have actually discussed this in this group before.
Yep.
That people use sometimes regressors because they get a better functional connectivity, but that functional connectivity could come from other things than neural connectivity. So my question is there a better way of, let's say, if we understood the causal connection better between let's say heart rate and bolt signal and respiratory signal and bolt signal and the global signal changes, that we could regress that better out, having actually a mechanistic model rather than, people typically regress it out with, component analysis. And do you think that understanding better the mechanisms would be helping?
Yeah, I mean, I think in the ideal world, if we had an ideal model, then we would need a lot of data, and we would have to do nonlinear fitting to the model, and let the model sort of split it into different components, and then analyze those underlying signals or hidden parameters inside that model. I think that regression isn't the right approach, because it's like the same problem people have like in ecology, right? You have this web of interactions, and if you split it, you've changed the whole system. And I think regression is useful for things where there's a huge effect of one thing, right? Or censoring, where you're just saying this data is totally corrupted, I can't use it, right? Then it makes a lot of sense. And in some cases, regression acts as censoring, as I've said before, because regression will typically fit to the largest, the most extreme values and get rid of those areas. And so in cases where you have extreme values, then regression and censoring approach each other. And so that's where it makes sense. Anything below that, I think that regression is either going to have negligible effect, or is going to, as what you said, lead to spurious inferences. Because when all these things, the problem is all these things are highly correlated, right? You have respiration, heart rate, activity, arousal states, your fMRI signal. There's just a lot of co-correlations between all these measurements. And so I do think we're limited in what we can do by splitting it into signal and noise arbitrarily. I do think ideally you would have a full model and fit it, but then you need a lot of data.
So you're suggesting probabilistic models, and I mean, if we had, let's say, all the relationships and the characteristics of the noises, then we could fit a whole model in a probabilistic sense.
Yeah, I haven't really thought about, it's
What's
right. Yeah, I suppose a probabilistic sense. Yeah, in some sense, you're just trying, it depends on, I suppose, right? The challenge would be to decide what you're trying to estimate, right? And so, whether you can sort of do some sort of, integrating across unknown parameters. And so I don't know the exact approach that would work. But I sort of, the sense is that, yeah, I suppose, for example, let's do a thought experiment. You could maybe say, what is the, so a very simple thing might be just to split into what is, like, if we could just, we might even start with simpler things, like what could we really say is local activity versus spatial temporal traveling wave activity. So we might even start with simpler things and sort of work our way down to more complicated models. So I think you're raising a lot of good questions. I don't know what the right answer is. It's just my intuition is that we need to, the general little model is great for what it does, but it is limited. But yeah, I don't have a good solution for what to do.
Jeff, go ahead.
Thank you, Tom. This has been awesome. I have 2 questions. One has to do with the spatial temporal issue you were showing. I've not an MEG expert, but I have worked in trying to put MEG and MRI data together. And I have seen from the MEG side some of that spatial temporal propagation, but on a time scale that is, in the millisecond range. So one of the questions that has always bugged me is that the fMRI time span we're talking about, anywhere from a half second if you're really aggressive with your equipment to up to about 2 seconds for a TR. It's hard for me to believe that the brain propagation wave is that slow in reality. I think it might be that we're measuring it every 2 seconds, and then we're sort of filling in the gaps. It's sort of like its own temporal filter. And the MEG data looks far more detailed and spatially specific. So I'm wondering if you've seen or have any comments about the temporal limitation that fMRI and the sense that we're looking at seconds instead of milliseconds here. And the second question is just if you have any comments about scanner noise or artifacts that are based on image artifacts from scanner badness, as we call it.
Yeah, okay, so thank you for your questions. So the first one is a really good question. So there definitely are different timescales for the traveling waves. I mean, there are traveling waves at millisecond timescales, and then what we're seeing in the fMRI data is much longer timescales, as you indicated. So what sort of lends us to believe that these are real is that there is both the papers I cited, these recent papers, also look at monkey electrophysiological data, and they showed found similar timescales. And there was a recent paper in bioRxiv that I believe is probably coming out for publication soon, also by the Penn State group, Xiaolu's group at Penn State, which looked at data from the Allen Brain Atlas, so NeuroPixel data. And I don't remember the details, but I also believe that he found similar timescales in sort of this electrophysiological data. So my guess is that the brain has multiple traveling waves at the same time, and there's probably the ones that go across long distances are probably lower frequency, and we don't know why they're there, but they may be due to more autonomic sort of nervous system sort of maintenance and the general arousal stuff. And then sort of the faster traveling waves tend to probably, I would guess, are probably more localized. There's a really nice review paper by Terry Sinowski and others in Nature Neuroscience Reviews about traveling waves. So that talks a little bit about this. And so I believe my current sort of impression is that there are these different traveling waves at different spatial and temporal scales, all happen simultaneously, which makes the problem even more interesting. Oh, and the second question is artifacts. I'll talk a little bit about that when I show the slides on the QA and QC. I didn't prepare a comprehensive view of that, but maybe I'll show what I have, and that can lead to further discussion.
Any other questions before we head to the QA QC part? Okay, fabulous.
Okay, let me just advance ahead. Okay, so some slides on QA and QC. So these are some slides I took from a book chapter we wrote. So I was part of the lucky enough to work with Gary Glover and others on the fBurn QA, and so we would that was a group that met for many years and met weekly to talk about QA and QC issues. And so I think most of you are familiar with the fBurn QA. There's all these different parameters. Pradeep did send me something. I think there was a note from Stephen Struther about different, there was a question about what parameters are what. So I'll go into that a little bit. This is sort of some notes that I made internally for ourselves. And one of the things that I think one of the questions was, you know, the relationship between SFNR and SNR. So at least the way I look at it is, you know, so the SNR is, you know, based on the subtraction of all the even images minus all the odd images, right? And so basically SNR should track SFNR when the noise is temporally independent. And so that is sort of the way we look at it. And so I do agree that SNR and SFNR tend to track each other fairly well. And I'll go into a little bit of sort of our experience with looking at SNR, SFNR, and how it might differ from RMS, and how you might use RMS, RDC, SFNR sort of together to sort of help debug or show you some examples of how we've used those different metrics to debug what's going on at our site. So as I said, we definitely do, we're sort of obsessive about QA at our center. So this was a case where we have, at our center, for a long time, we had 2 scanners by the same manufacturer. Now we have 1 from each manufacturer, but essentially you can sort of see this was over, you know, a year of tracking of data. And so we would, for this scanner, this was when this QA stopped here because the scanner was being upgraded at that time, I think. I'm not sure if that's the case. Yeah, that's probably the case when it's being upgraded. This is back in 2010, 2011. But you can sort of see we sort of track all these things fairly carefully. In addition to these measurements from the fBurn, we will also grab as much information as we can about the frequency, center frequency, transmit gain, receive gains, everything we can about the system status and what was being used. And so those things can also be useful for debugging. So let me just go through an example that's in this book chapter, which this was happened when scanner 1 was being upgraded. And when it was first installed, you can see here that scanner 2 was the scanner that was there. And typically on this was a GMR750. We had RMS values around 0.04, and that's what sort of we used to. And in fact, the specification we told the manufacturer that we had negotiated when we bought these scanners was you have to keep the RMS below this red line, and you have to keep the SFNR above this red line, and so on and so forth. So we had contractual sort of acceptance criteria on our scanner, and then anytime it deviated from those, we could tell the manufacturer, hey, you need to come and fix it. So they were sort of contractually bound to get, so when the scanner first came in, you can see the RMS values are not too bad, but you know, they're above the 0.06 threshold. And the question is, how do we go from here down to here, right? And so when I looked at this data, you know, so in this period, I look at the data and say, okay, the RMS values are much higher. SFNR is pretty much the same though, right? I mean, there are some variations from day to day, and sometimes this is just, sometimes positioning of the phantom and stuff can have an effect, but you know, it's not, it's passing. But what's also failing is RDC, right? RDC is much lower here in the scanner, and then it comes back after they fix the problem, right? So these 3 things together are sort of what I typically use as a first stab at trying to diagnose a problem. And so in this case, the RDC being low means that there's a lot of spatial correlation in the data, right? And then the SFNR being low means that the temporal stability on a per voxel basis is pretty good, right? But the RMS being low means that when you're averaging, the RMS is an average of the ROI, so you're not getting the benefits of averaging, right, independent samples to bring that RMS level down to a lower level. So this means that you're looking for a system component that is causing spatially sort of, that has low frequency effects on your signal. And one of the things that from experience we know that is the general culprit for that is problems in the transmit amplifier, because any sort of variations in that transmit amplifier affect the whole ROI, right? So the stability can be okay on a per voxel basis, but it's so, because things are varying with time, there's a spatial coherence to the noise. And so, in fact, there was a problem with the transient amplifier. We basically told the manufacturer, you have a problem with your transient amplifier, come replace it. They replaced it, and lo and behold, that fixed the problem. So this was a nice example of that. This is another example. I don't believe it's from our site. It's probably from one of the efferent sites where they were looking at, once again, they had a really big problem with their transient amplifier. So they went from 400 to 100. So in this case, the difference is so big that it's basically, you don't need too much detective work. And in this case, they just replaced the RF amplifier and it went back. So in this case, probably all the metrics would have gone down. And in this case, a lot of, as you know, a lot of, when you're dealing with these very complicated systems, really only the first level of approach is always just to swap components, right? And so the game you're playing is, what's the cheap, how can you swap components in a way that doesn't cost a lot of time and money? RF amps are fairly easy to swap out. Gradient amps a little more issue. And then obviously if you have to replace the gradient coil or the RF body coil, that's much more expensive. So manufacturers won't want to do that too easily. But they're generally happy to replace an RF amp. And those tend to have issues quite a bit. This is just showing that, in the efferent that we found that SFNR was, this is sort of a subject resting state map from a subject that had low SFNR and one that had high SFNR. And in those same subjects, it's clear if you look at the data that the SFNR is really driven by just, lots of different effects happening. And so not, as I said before, I think one of the issues is when you have these really big sort of swings, then that really totally dominates your functional connectivity maps. And so anything, my guess is even if you'd sort of censored out these regions, you could have recovered some bit of this functional connectivity map. It wouldn't be necessary to totally clean up the data. And I think in that chapter, we also showed that if you looked at the high SFNR data, you get much better effect sizes than you look at the SFNR quartile. So in terms of QC, in terms of, and this was I believe, in vivo SFNR. So in terms of, now we're talking about sort of QC, then, obviously segregating data into high and low data quality seems to have an effect. And then this was just some data we were looking at. I believe it's, we've sort of masked the sites here but the point we want to get across here is we're looking at RMS and SFNR, and we're plotting the data as violin plots, and you sort of see the whole distribution for every site. And I think what I've seen in terms of looking at this sort of data is each site tends to have its own signature. It's sort of, once it's at a certain value, it sort of stays there, and all you're really looking for is, does it deviate from its sort of bubble? Now, if you have a site like this, which is sort of deviating from sort of the mean, the interesting thing is oftentimes it will deviate in one parameter, but be pretty good in another parameter. Although in this case, it's low on this and high on this. So this agrees. And so for this site, the conundrum that multi-site studies always have is, how much effort do they actually want to put into, fixing a site and bringing it into line? And it's even more complicated when you have like Siemens, GE, Philips, Canon, and those tend to actually group into different sort of segments of these metrics. And so that is an issue with multi-site QC. And so I know in the afburn, we did spend a lot of time looking at some of these. We found that, for example, in that study, the difference between the fat saturation and water sort of excitation, sort of the pulse. So the GE uses a spatial spectral pulse. So it sort of just tries to excite water, whereas, and it has a very complicated spatial spectral pulse, whereas at that time, the Siemens was using a fairly simple, I believe, binomial water excite pulse. And so the difference in slice profiles of those two actually led to a big difference in sort of the phantom measures. But even just that very simple observation, because you sometimes access these scanners is very little, and you're trying to get data from different places. Even with people like Gary Glover and lots of really smart people on the call, that still took months to figure out. And so I think for multi-site study, we don't often have the luxury of doing that. And so often what I see is, as long as the human data looks reasonable, that these big studies are just sort of, you sort of accept what you have, and then you just sort of make sure that each site doesn't go out of its own sort of comfort zone. In our own experience, unless something is really off, it's very, very difficult to move something, like even to move something from here to here, it's not necessarily trivial. And there's no guarantee that you can do it, you know, and it's almost a luck of the draw in terms of, you know, even if you, and we actually did this, was sort of in one instance, we actually told the manufacturer, we want this, and said, okay, we're going to pick the best component at the component level. We're going to pick all these things to be the best, you know, like the best gradient, the best gradient amplifiers. So even if you do that, once you assemble it on site, you know, depending, the interactions are just still not well enough understood that, you know, even when it's somewhere on site, you can have the best components, but the interaction of those components can conspire to give you something that is not necessarily the best performing system. And so in some sense, maybe a random approach might be better. Just don't even, you know, just take random components and assemble them together and hope that random cancellation helps you. So in our case, we sort of felt very confident that, you know, we would get the best system we could, but you know, it turns out that these systems are so complicated that it's not as straightforward as that. Okay. So that's really all I have to say as a QA, QC. My perspective is really from the point of someone who's directing a center and sort of making sure that we, you know, can make sure our systems, you know, are performing as well as they can. In addition to these QCs, we do have things where we will have scans sent from like example images from every scan that's taken that will go to our technical team, and we have some software to sort of, you know, just highlight if something looks really off, like if there's an obvious artifact or something. Just not only to deal with system issues, which are typically not going to be a problem if the scanner has passed the QA that we do every morning, but more also operator issues. You know, oftentimes we find that the QA QC issue is really just a little mistake is entered to the protocol. You know, someone has changed something and then it just keeps propagating. And then what really kills us is when a group has gathered data and then comes back a year later and says, our data are terrible, what can you do? And at that point, it's too late. That's sort of my perspective on QA and QC. We definitely, you know, what we tell everyone is you've got to look at the data right away. You know, you don't need anything sophisticated necessarily to get, you know, you're really just trying to protect yourself against the really big disasters, right? Those are the things that really will kill a project. If your data is sort of okay and it has some issues, you can generally salvage it. But if you've really just collected the wrong data, or all the data are just like zeros or something, you know, there's no way to salvage it. So I think the actual QA and QC procedure doesn't matter as much as whether people just do something.
Can I ask you a quick question? So related to this, so you had a very smart contract where you said, like, if the QC measures are below a certain thing or above a certain value,
Yes.
then they have to bring it back. Because if I had a scanner that was 3 times worse than any other scanner, you know, I would want a different scanner. I mean, it seems enormous, like differences in the. I was really stunned by some of these outliers in your plot. I mean, if you get a contract like what you do, right? I just get a new scanner.
How did you make that happen? Did you use a gun?
Well, that thing was done. That was done like that negotiation. The one where we had the thing built in was probably done 15 or 16 years ago, and things were very different back then. And you know, and it always depends on which vendor is more desperate and what you can get. You know, I can say we tried again more recently, and we weren't able to get something like that. and then the question is. The problem is, you know, you can be like, you know, how well the F-burn corresponds to the in vivo performance is still not well known, right? I mean, those F-burn things are fairly tight. So whether you have 0.04 versus 0.08 in RMS frankly, I don't think it probably makes that much of a difference in the quality of the data, given all the other sources of variability. And other things, for example, like ghosting levels, right? Ghosting levels, especially for multiband or other things, those can be like on a FR and agar phantom. What we found is, those can look horrible on a phantom, right? Because you have this very uniform object, and all the ghosting will be very coherent and look terrible. But then you go scan a subject, it's like, oh, that's not so bad. It's like, it's no, and for example, ghosting levels differ quite a bit from, in our experience, from vendor to vendor. And yet there are, very successful studies that are required on scans where if you look at the ghosting levels on a phantom, they're horrible. But, those are still, people are still acquiring lots of data on those scanners. And I think the data is actually probably in vivo. My guess is the in vivo data is probably pretty good, right? So I'm not sure I answered your question, but I think there's also a huge cost. If I have a scanner that I think will be okay, even if it's not passing all the specifications, then you have to make a decision. Do you really want to go down for a month? And then maybe there's no guarantee the next scanner is going to be better. It's really a 50-50 flip of the coin with these scanners. So I think all you can do is do mitigation issues. If the manufacturer is willing to work with you, then do little part swaps here and there. I mean, the most they can do is pull out the gradient call and put a new one in. And beyond that, there's really, I mean, there's either that or I know in some cases there's been sighting issues where, for example, they have to create better stability for the scanner. So I know one site that was a solution. But if it's these really subtle issues where it's sort of marginal, especially when it's a marginal fail, then it's probably not worth it to do stuff. If it's really way out and you can, and I think if you can show it in vivo data, then yeah, it would make sense. And there are real limits, right? But generally, the things that are really failing are easy to fix, right? It's the things that are sort of close that are very hard to fix. And close can be even a factor of 2, right? Because if your RMS is 0.04 to 0.08, going from 0.0 to 0.04 is not necessarily trivial. That's been our experience, at least.
I have a question. So do you see any reasons to update the FBurn QA guidelines?
Well, I mean, I think you mean in terms of the metrics that are measured or the protocol?
Guidelines in general, yeah, protocol, so to say.
Yeah, so I mean, I think, obviously as things have changed, right? I mean, the original FBurn is done with a single band acquisition. And so, for example, I was part of the initial QA group for the ABCD consortium, and there what we did was we do both a multiband acquisition using whatever their scan is going to be, and then we use the single band FBurn as sort of something that we had more history on. So I do think that, yeah, studies have to be sort of flexible and potentially adopt additional metrics that are more pertinent to their study. And so, for example, the ABCD data acquires both single band and multiband as part of their QA process. Whether the metric, and I think there's certainly room for new metrics, but I think that the metrics that are probably cover most issues, and anything else would just sort of be a second order effect. Just to even measure those things gives you a certain feel for things. I think one thing that is sort of would be interesting to sort of look at is, the FBurn is really mostly focused on fMRI, right, and temporal stability, and whether there should be something for like diffusion MRI because it's so prevalent now. But when we looked at it, it wasn't clear what you could do very quickly and how to analyze it very quickly. So so I do think that there's certainly room for improvement, but I think the fact of the matter is that what drives a lot of the process is whether the QA is even done or not, and how well it's done. I mean, very simple things like, is the phantom secured well? Is it stationed well? Is the same person doing it? Just little things like that can have a big effect. I mean, you'll see big changes in metrics when, like, one student graduates and then the student comes. I mean, that can have a much bigger effect than your system sometimes.
Thank you. Jeff, go ahead.
Thanks, Tom. So getting back to my question about noise, the question that I am most curious about is the graph you showed with sort of like the spatial distribution and how the transmitter affected sort of some central slices, but the outer slices looked fine. And sometimes we see in some resting state data the reverse being true. And so I'm curious about your comments, because usually what happens is you see one bad slice or two by some detection method, and then you just throw out the whole volume, or you might throw out surrounding volumes. And I mean, I'm assuming we're saying, for the sake of the question, we've already know that motion is taken care of. And so we're just looking at other things besides motion that would throw up like a few slices that might look bad. And then we throw out the volume. Is there any slice specific methods or areas that you're investigating to rectify any of those individual slice type issues without throwing out massive numbers of volumes?
Right, so let me just make, so one thing is that all the efferent stuff just looks at the central slice, so those metrics are not sensitive to that. But I think what you're, just so I understand what you're saying, is you're saying that you'll gather data and you'll just find that certain slices are just worse than others in vivo data, or?
Well, what we see is looking at large data sets that some subjects in some sites, once you've taken care of motion, there'll be just some bad slices, and it's not even clear that it's scanner artifact versus something else that might be causing it, or reconstruction artifact. But we're just getting some focal, randomly distributed throughout a volume over like hundreds of volumes, some bad slices that don't show up in the volume identified as motion. And so what do you do with those individual slices if you don't want to throw out volume after volume of one or two bad things? And I'll say it in the diffusion world, FSL has a great tool to try to fill in one or two bad slices when it has these huge sort of like the susceptibility artifacts, where you'll see this black hole appear when there's something going on in just the right part of K-space. And so then that slice goes blank, and there's a way of sort of interpolating between slices and coming up with a rough estimate. But diffusion data has a lot of other things going for it to do that. So I'm trying to say that's diffusion. Now we're talking about resting state and fMRI data. When you have individual slice data, how do you handle not throwing out large parts of the baby just because one little piece of the bathwater looks sketchy?
Right, and is this multiband or single band data?
Mostly single band, but there is some multiband, because it's obviously a multi-site. So there's some spiral in there. There are even some where the phase encode direction is an AP for the fMRI. Don't ask me, I can't explain that. But it's stuff like that.
Yeah, so I mean, I think the solution, I mean, I haven't really dealt with this specific problem, but as you're talking, just, the realm of solutions would depend a little bit on whether you have access to the K-space data or not, right? So if you wanted to go all the way, obviously, what you could do is, there's these low rank sort of reconstruction methods that have been developed, for example, by Mark Chu at Oxford, where, if you look at all the data that's acquired over time in K-space, there's a lot of redundancy in that data, both across space and time. And so if you really want to fix something and you had all that K-space data, you could sort of do some censoring of K-space data and then just sort of do a low rank reconstruction of spatial temporal reconstruction of the whole data set. Similar to how like Netflix fills in your preferences for movies you haven't watched, right? I mean, it sort of looks at the structure of who likes what movies. Beyond that, I mean, I think other approaches would include, I don't think there's any easy solution. I mean, you could do some, obviously some censoring, slice-specific censoring, and include that in your general linear model somehow, right? Or else you could do some, if you just have the image data and you feel like it's just missing at specific time points, I mean, I think you could do some spatial temporal interpolation even in image space, and there is a lot of smoothness in the data both spatially and temporally, and so I think it seems like it would be, the solution would be specific, but I don't have any experience specifically with that type of problem.
Thank you.
Sure.
Great, thank you so much. Any other questions or comments, Joe? And we have some people from France too today. Yeah, so if there is no other questions, let's thank Dr. Liu for his generous time and sharing his, this has been a fantastic talk. I looked up like 10 papers to read,
Great,
so
great.
thank you so much. It was very helpful, and yeah, I'll keep in touch.
Great, super.
Thanks everyone for joining.
Yeah, thank you for inviting me, and this is a great initiative you have, and I look forward to keeping track of what you guys are doing.
Thank you.
Great. Thank you.
Take care.
Okay. Have a
Bye-bye.
good day. Bye.